Welcome everyone to the last session, and we have 4 speakers, and Kai start with talking about integrated ChIP-seq data analysis workshop.
Okay, hello everyone, my name is Kai. Thank you very much for having me this year. Actually, this is my first year to attend to this bioconductor meeting. Thank you again for attending this workshop titled Integrated ChIP-seq Data Analysis Workshop from FastQ to Plots. So before we get started, you can choose to run the workshop. I think you're already very familiar with the procedures. You can either choose to run the workshop on the cloud by clicking this link here. I've already posted the passcode and username via the chat box, so you can check out the username and password there. Alternatively, you can also choose to run the workshop locally by downloading a Docker image. The Docker image contains all the essential softwares essential for this workshop. So you can just Docker pull and run the Docker image, and you're all set to run the workshop. And in order to figure out the more details, instructions on how to run Docker image, you can go to my GitHub page here, the slides via the chat box. So you can check out the chat box and download the PPT slides there. You can copy and paste the URL and all the codes from there, so you don't have to type in. So here is a brief outline of the workshop that we are gonna give today. So first of all, I will give a very high-level introduction to ChIP-seq analysis. Basically, I'll talk about the basic concepts behind ChIP-seq. I will also talk about the commonly used tools for like quality control, read mapping, as well as peak coding procedures. And the focus of this workshop is on the downstream analysis. Therefore, for the demonstration part, I'm gonna talk about mainly 2 R packages for the visualization and data analysis for downstream ChIP-seq analysis. And one package is called ChIP-seq. The other package is called TrackViewer. And both of the 2 packages were mainly developed by Jianhong and Julie. And they are also the co-authors of this workshop. And of course, at the end of the workshop, we're going to answer your questions. And feel free to post your questions via the poll. So, like I said, this workshop is about ChIP-seq. So what is ChIP-seq? As you can see from this diagram here, it consists of 2 consecutive steps, ChIP and seq. ChIP here represents for the chromatin immuno-precipitation. Basically, it is a strategy to pull down the DNA interacting fragments. And seq here simply represents for high throughput sequencing. And this strategy has been was first proposed back in 2007 and quickly supplanted the ancestor ChIP-seq strategy. And ChIP-seq has been widely adopted by biologists over the last almost 2 decades to analyze the DNA binding sites for interacting proteins, as well as to analyze the genome-wide landscape of histone modifications. And ChIP-seq, when combined with other type of data like RNA-seq data, can also be used to infer the gene regulation networks. As with all other techniques, ChIP-seq is far from perfect. There are a lot of problems with ChIP-seq. And one of the big problems that I think is about the high false positive rates. So therefore, to ensure you have the best outcome from ChIP-seq, when you design your experiment, I suggest you to follow the suggestions offered by the ENCODE project. So here I listed several suggestions that are suggested by ENCODE projects. So basically, first of all, you have to have a so-called ChIP-seq grade antibody, which have very high affinity to the binding protein, binding target. And you should have at least 2 biological replicates for your analysis. And also don't forget to prepare a matching controls for your sample. In terms of the sequencing depths, it really depends on your analysis. Typically for the transcription factor ChIP-seq analysis, you would require 15 million reads. But for histone modification ChIP-seq data analysis, 30 million reads is typically considered as the minimum. And here is another super simplified version of ChIP-seq data analysis workflow. So basically the raw data that you receive from the sequencing companies are typically in FASTQ format. So the first thing I'm gonna do when you receive the FASTQ file is to perform some read mapping procedure to map those short reads onto the reference genome and create a band file. And then after you have the band file, you're gonna run some peak color steps to generate the peak information which are stored in the bed format. And of course, as you can see from this workflow, there are multiple quality control steps throughout the analysis. And there are a lot of tools for quality control in the market. So to name a few, MasterQC and DeepTools are two of the most famous tools that people commonly use. Like I mentioned before, the focus of this workshop will be on the downstream analysis. Therefore, in the demonstration part, I'll talk about ChIP-seq Anno and TrackViewer, these two packages. But before I dive into the downstream analysis, I still want to talk a little bit more about the upstream analysis, especially the mapping and the decoding procedures. So for the mapping procedures, over the years, like in the past 2 decades, people have developed over 90 different mappers. As you can see here is the different mappers that are developed over the years. And these mappers can be roughly divided into 2 categories according to their underlying algorithms. And as you can imagine, there are a lot of review articles that compares different aspects of the mappers. So here we would recommend using BAA, the Bidirectional aligner, to perform the mapping step. This is because that we think the BAA has a really good compromise between the mapping precision, computational time, and many other parameters. But when it comes to the pick color, the situation is a little bit more complicated because depending on the binding mode of your target protein, the tag density profiles can differ dramatically from each other. As you can see here from this example, for transcription factors, a typical peak will be very sharp and tall, as shown here. Whereas for the histone modification ChIP-seq peaks, these peaks are usually very broad. So it is very challenging for a single peak color to accurately assign peaks for different scenarios. And in reality, different peak colors are specifically optimized for different peak types. And again, there are a lot of tools out there for peak calling steps. For narrow peaks, we would recommend using Next2 for a peak color step. But for the broader peaks, we would recommend using Cedar for the peak calling step. And after you perform the peak calling step, the called peaks will be stored in the bed format. A bed format is simply a test format, which stores the peak calling information. So as you can see here, basically each row here in the bed file contains the information of a specific peak. So as you can see here, there are different variants of bed files, depends on the specific peak colors that you use. So for a minimum bed file, there are 3 fields that are required, namely the chrome star, chrome end, because these 3 columns are essential to document the position of the peak. And you do have to look into the documentations for different peak colors in order to figure out what other columns represents. So now that we have the bed file, the next step would be to perform downstream analysis. So to this end, depending on your specific research goal, there are a lot of downstream analysis that you can perform. So typically, the first thing people would do is to annotate the peaks. And it is also very common for people to perform like Metagene analysis, overlapping analysis, Motif analysis. And usually when you are done with annotation of the peaks, you still want to perform like enrichment analysis and gene regulatory network analysis to figure out what you're gonna do next. So here, I'm trying to summarize all the commonly practiced tasks into 14 examples. So I will talk about the first 8 examples that are achieved with a ChIP-seq Anno package. And after that, Jianhong will take over and talk about the TrackViewer, which covers the next 6 examples. So now we can move on to the demonstration part. So for this part, you can go to the vignette, where I also share the link to the vignette where the chat box. You can just go to the chat box, copy and paste the vignette URL, and open the vignette. From the vignette, you can copy and paste the code from the vignette and try to run by yourself. So this vignette is pretty comprehensive, which contains a lot more details than the demonstration here, because I just want to make sure we have enough time. So I use a simplified version of the vignette. Okay, so probably the most important downstream analysis is peak annotation. So first of all, I'm going to talk about how to use ChIP-seq to perform peak annotations. So basically, to annotate a peak, you need to perform these 4 consecutive steps. So first of all, we have to read in the peak data, which is in bed format, and convert it into a G-ranges object. And second, you have to also input the annotation data, also with to G-range function, and convert it into a G-range object. After you have both the peak file and annotation file in the G-ranges object, you're going to use annotation peak in batch function to annotate, to build the connections between the peak and annotation. And this step can be achieved with the annotate peak in batch function. And finally, you can also choose to add additional informations to the annotation file by using the adding IDs function. So move on to the first step. So first step is to reading the peak file. So here, I'm trying to use the example data that is stored in the ChIP-seq package. So you can use the system.file command to locate the path of the example file that is stored in the package. You can also choose to replace the path variable with your own customized set so that you can run your own set instead of using example data stored in the ChIP-seq package. And then you can use the 2G-ranges function to convert the bed file peak into G-ranges object. So here is the result. As you can see that the peak file in bed format has already been converted successfully into the G-ranges object. So after we have the peak file in the G-range object, the next step is to read in the annotation files and convert it into G-range objects as well. So as you know that there are multiple different sources of annotation files. And for example, you can choose to use the ensemble annotation files. You can also choose to use like TXDB, UCSC annotation files. And here for demonstration, we use the UCSC annotation files. Use the same 2G-ranges function. You can convert the annotation files into a G-range object as shown here. So after the first 2 steps, we have both the peak file and the annotation files in G-range objects. And then we're going to use annotate peak in batch function to try to build the connections between the annotation and the peak. Actually, there are many more arguments for this function that you can use to tune the way how you're going to annotate the peak. So here, I'm only using the default parameter. So we can look into the documentation to see like how many different other parameters you can tune for the annotation process. And here is the result. The result is also a G-range object. As you can see from the first column to the peak column, these regions represent the peak information, whereas from the feature column to the last column represents annotation information that has been already linked to the peak. So because, like I said, here we use the TXDB annotation, the ID here under the feature column represents the entrance here because I just use the default. So the default is the nearest location here. And finally, you can also choose to add additional informations by using adding ID formula. In this case, one caveat is that when you are using the TXDB annotations, you have to use entrance ID for the feature ID type. If you are using ensemble annotation files, here you have to replace the entrance ID with the ensemble gene ID. And yeah, this command is also very straightforward. You just apply the annotation files onto the annotation files. And after this command line, you can see that you have successfully added another extra column, which is called symbol, onto the annotation file. So basically this 4-step, after this 4th step, you can successfully annotate the peaks with ChIP-seq. I think it's pretty straightforward and simple to use. The second example is about finding overlap peaks for biological replicates. So the same thing that it's very similar that first of all, you have to read in your peak files using the 2G-ranges function. Here is a peak 1 file, here is a peak 2 file. Here you can notice that the peak 2 file is in GFF format. So typically a peak file will not be represented in the GFF format. Here we just want to show that the 2G-ranges object function can also be used to deal with the GFF format, just to show you that it's capable to do so. And after that, you can use the find overlapped peaks and input the 2 peak list. And you can get the result is also in the G-range object. So as you can see, the first overlapped peak, it actually comes from 3 peaks. One is from the GR1 this side, and 2 are from the GR2 this side. So you can also choose to make a Venn diagram to show the overlapped peaks, like between different data sites. Just with the make Venn diagram command, here you can see we successfully created a Venn diagram to visualize the overlap peaks. Another important question that people ask is, like, how do I select the features to annotate? In order to answer this question, you can leverage the assign chromosome region function to try to visualize the distribution of the binding site across different features. Like, for example, here we use assignment chromosome region, and on the overlapped peak that we called in the previous example, and here is the result shown here. You can see that most of the peaks are assigned to promoter regions. And as you can imagine, it is possible for a single peak to span multiple features. So under such scenarios, you may ask, like, which feature should I assign peak to? Under such scenarios, you can use the precedence argument to try to determine the order of annotations. For example, in this case, the promoter is at the very beginning of this list. Therefore, if a specific peak spans both the promoter and other features, like maybe UTR, the peak will only be assigned to the promoter instead of UTR. That's the function of the precedence.
I want to add, so there's a nucleotide level. If you just choose, then a peak could be assigned to multiple places. Nucleotide level?
Yeah. If you said, actually there are 2 different representations. One is the peak-centric view, the other is the nucleotide-centric view, right? So if you assign the nucleotide level to true, you can assign the peak to all features, right? Okay, so the next example is to try to find peaks that overlaps with bidirectional promoters. So bidirectional promoters are basically refers to some genomic locations that are between the transcription start sites of 2 adjacent genes that are transcribed in opposite directions. So the bidirectional promoters usually regulate both genes on the opposite direction. And in order to find the bidirectional promoters, you can leverage the peak near BDP function here. It's also only one command, and you can figure out which peaks are with overlapped bidirectional promoters. And example 5 is about motif analysis. So basically what we want to achieve here is trying to get a summary of peak consensus. To achieve such goal, actually, we have a lot of alternative methods. For example, you can use the get all peak sequence module from the ChIP-seq package to try to output a FASTQ file, and then use some other tools like Meme or Homer to try to perform the motif search and motif visualization. Alternatively, you can also use summarize pattern in peaks function here, also in the ChIP-seq to try to test the predefined patterns and see if the target patterns are enriched or not. And here in this workshop, I'm going to show you how to use oligo summary function to calculate the z-scores for all different potential oligonucleotides at a given length, and then try to visualize with multiple stack. Okay, so first thing, you still have to get the FASTQ sequence using the get all peak sequence. Just apply this function to the overlapped peaks object, and you get the FASTQ files saved in the seq object variable. And then you can try to summarize the FASTQ motif information using the oligo summary function. So, and then you can use motif stack to try to visualize the motifs that are contained in the FASTQ format, FASTQ file. The result can be shown here. And another very common task that people are gonna try after you annotate the peaks is trying to perform some enrichment analysis. For such purpose, we have provided another 2 very helpful functions. One is called get enriched goal, the other called get enriched path. So these 2 functions are also very straightforward to use. You just apply get enriched onto the overlapped annotation file, and you can see the enriched terms. One thing that I want to point out is that for the get enriched pathway function, you do have to provide the reactome.db as the parameter here. We have multiple sets of peaks. It can also be very interesting to see whether those peaks are correlated to each other. So in order to see whether the peaks are correlated to each other, you can use some statistical tests to achieve such goal. For example, you can use the hypergeometric test that comes with the make Venn diagram function to perform this analysis. However, for hypergeometric test, you will have to estimate the total number of potential binding sites, which can be very challenging and hard for a lot of biologists. Therefore, we also developed another permutation test. And with permutation tests, you don't have to estimate the potential total binding number ahead of time, and just apply permutation tests, and you can see whether the peak are significantly correlated to each other or not. So before you perform the permutation test, the first thing you have to do, you can choose to remove the hotspots by using this function here. And after that, you still have to download the pool information from, usually you can get the pool information from the ENCODE project. The pool is to sample the data. And after that, you can just apply the peak perm test on different sets of peak list. And here is the example output. On the left, the black line represents the expected permutation test value. And the green line here represents the observed permutation test value. As you can see that the distance between these two lines is pretty large. Therefore, we have a very huge Z-score. The last example I'm gonna talk about is about Metagene analysis. And we have multiple sites of like peak files for different transcription factors. You wanna visualize the binding patterns. So under such a scenario, you can try to use the feature aligned heat map function to visualize the metagene for different transcription factor peaks. Before you do this, you first need to extract the signal from the peak list using feature aligned signal function. And after that, you can just apply the feature aligned heat map, and it will generate the very beautiful metagene heat map for different sets of transcription factors. And alternatively, you can also choose to profile the same information with feature aligned distribution function. This basically contains the same information, just in a different representation. So I think I finished the ChIP-seq annual demonstration. So next, I will give it to Jianhong to talk about the track viewer. I hope that you have enough time to cover track viewer, because I know it's also a big, very huge package.
I'm Jianhong. Today I will introduce track viewer, a tool to do lollipop and dandelion plots for methylation and mutation data. Epigenetics is the study of a mechanism that produce phenotype changes without alternations in DNA sequence. It includes DNA methylation, histone modification, and non-coding RNAs. There are multiple types of DNA methylation. For example, 5-MC is a methylation of the 5 position of a cytosine. The DNA methylation can control transcriptional expression, like a signal for a switch trigger. Usually methylated DNA will recruit some protein complex to repress the binding of RNA polymerase or TFs, which in turn repress the gene expression. For example, if the promoter region of the tumor suppressor gene is methylated, as shown as a hypermethylation here, the expression of tumor suppressed gene will be downregulated, which in turn lead cancer happen. Like UCSC Genome Browser, Washington University provide epigenome browser. It is very similar to UCSC Genome Browser. It can also show the interaction in different chromatin regions for ChIP-seq. Another type of data I want to involve is site mutation. Mutation is basically an induced misspelling of a DNA sequence. There are several types of them: point mutation, chromosome mutation, and copy number variations. For chromosome mutations, there are deletion, duplication, inversion, substitution, translocation. For point mutations, there are SNPs and insertion and deletion we call indels. For human, Cosmic database is an online genomic browser to view the point mutation, and DB-SNP contains not only human single nucleotide variation, but also other species. Beyond the online tools, there are many tools to create a mutation diagram, methylation mutations, and the single nucleotide polymorphisms data. Require a special type of plot called lollipop or needle plots. The most famous two are C-Portal mutation mapper and PFAM lollipops. There are so many choices why check viewer for lollipop, just because it is very beauty. It is minimized the design, and it's very simple, clean for publication, and it is very powerful to handle high density of data. The shortage for this tool is that you must be familiar with R, and there is no embed database. That is to say, you need to prepare all the data for plot by yourself. VCF file, BAD file, and their variations can be used as input file for plots. They are the most popular file formats to hold methylation and mutation data. To extract the annotations, you can use TXDB or EnzymeDB or Annotation Hub data and other online resources. By combining the annotations and methylation or mutation data, TrackViewer can plot the lollipop plot for low dense events and dandelion plot for high dense events. TrackViewer can also combine the coverage plot with the lollipop plot to generate the integrated plot. For lollipop plot, it is highly customized. For example, you can customize the label, the symbol, the color, opacity, and the size. You can also plot the score in stack symbol or pie chart, or even the shape defined by yourself. In addition, we implemented a web application of TrackViewer leveraging Shining package. The web application of TrackViewer is available at this web page. Today, I will not show the app. You can get more information from the videos in our publication. Here are some examples for publications that people using TrackViewer to show their data. In left figure, the author are comparing the missense variants in the region of ACE2 from male to female. In the right figure, the author are using the lollipop plot in pie stack layout to represent SNPs in different patients. In left figure, you can see the node color and the indicate missense tab. There is a count number in the node. It contains lots of details, but not busy. In the right figure, the author using color to show the base for each patient. We can go to the vignettes. Here is the examples for the vignettes. You can go get the vignettes in this web web page. We can also use view the vignettes in the Docker. So you just say type the address. You will get the documentation. You can just select the articles to get the reference here. So I will maximize the tool. When we go to the create lollipop plot, there are so many software. This is a table to introduce the software which can produce lollipop plots. To generate a simple lollipop plots, there are several steps. First, prepare the mutation data or methylation data, and the second is prepare the gene or protein model, and then do plot. Both of these two steps are G-ranges. You can see here is the simulation data. We simulate there are so many SNPs, and we just put the SNPs as a G-range data. We use a score to indicate the lollipop height, and use color to fill the lollipop, and this border is used to design the color of the lollipops. And for the futures, same, just use a G-range object. There are 3 of them. There are the start position and the end position. You just define start position, end position, and the fill color, and the height for each of them, and then use lollipop to plot them. After that, you can, here is an example to import VCF data in track viewer. It's same. We use a G model from TXDB to extract annotation data, and then you can plot it by lollipop plots. And you can also plot methylation data from a bed file. Just import bed file, whatever, from our track layer or other software. You read in, prepare it as a G-range, and then prepare the gene model, and set the color, the symbol, something like that, the plot tab. Here we set the plot tab as a pi. So you can see this is for methylation data. You can plot it as methylated and unmethylated. Here we also show we can plot multiple layers of the transcripts. So you just set a future layer ID as you want to set the layer. And also we can, here is an example to show how to use a PyStack layout to plot the methylation data for multiple patients. As I introduced in the paper, they are using the PyStack to plot mutations for different patients. You can also use we call the spider caterpillar layout, which just assign additional metadata in your SNPs, in your G-ranges. And we define this column name as SNPs side ID. You can put top and bottom. If you assign it as the top, it will plot the lollipop above the transcripts, or bottom is under the transcripts. if there are thousands of SNPs or methylation data, you want to show in a small size figure, you may want to try dandelion plot. This one is using fun style is fun. Then we plot for methylation data as looks like a dandelion. The thing is that please note that the height of the dandelion indicates the density of the events and not the methylation rate of the events. So I can give, as I introduced, the track viewer can generate the lollipop plot to show the methylation on SNP mutation status. It can visualize the regular race coverage tracks. It can be very easily integrated into standard analysis pipeline for various high throughput sequencing datasets, such as ChIP-seq, RNA-seq, methylation-seq, or DNA-seq. And it can produce a highly customized figure for publication. I would like to thank Hu Kai, Julia Zhu. I would like to thank Amy, and I would like to thank the Regeneration Next and the Bioconductor 2020 committee for giving me the chance to present this package. Thank you.
So basically, there are two questions about the ChIP-seq annual packages. First one is like how the function decides the belongingness of peaks which overlap to more than one genomic region. Jianhong, please correct me if I'm wrong. So I think in the annotated peak in batch function, there is a parameter that you can tune to try to like assign the features that you want to annotate to. So am I right, Jianhong?
Yes. If more than one genomic region, as I said, by default, it will just assign it into multiple genomic region unless you assign the parameter, say, we will consider exon first or 5UTR first, and there is a parameter to assign the order of them. So once you assign that one, then it will only assign one genomic region depend on your the order you assign in the function.
Okay. So for the second question, how to deal with a peak being annotated with multiple genes? I'm not quite sure I understand this.
It depends on your purpose. There are indeed in the annotated peak in batch, there are multiple parameters to make sure you can get the correct annotation depending on your experiment. For example, you can use find the nearest annotations. That will only output the nearest annotations, annotated features for you. There are also, you can use by overlaps. If there are multiple regions, you can multiple features by the overlap gaps. It will output multiple regions. Also, you can set as both of them. For example, if in the design max gap you cannot find any annotations, then it will find the nearest annotations for you. And also there are other methods, for example, find both bidirectional promoters or find find the both ends the nearest features by both directions. There are a lot of parameters you can assign for the annotated peak in batch. You can read the documentation for that. It will show a lot of examples for you.
Also, there is stranders. You can do stranded assignment. If you're only interested in, let's say, the same strand, the annotational strand, you can do that too. And so, like Jianhong said, Jianhong pretty much, said everything. I just want to make sure that's, you know, that's because some people ask this question on the web, think that ChIP-seq cannot deal with strandness. And actually it can. And also the peak start, peak end, you know, peak center, and you know, the feature start, feature end, peak middle. You can pick all these different parameters setting. And so usually people interested in nearest, but some people are interested in any genes, let's say, any features in the region of 5 kb or 10 kb, and then you will use an overlap. But like Jianhong said, if you know within 5 kb there's no gene, you might miss something. You maybe want to include in both, so both nearest and overlap to include all the genes. You can filter later using the output the data. For example, if you want to keep, you know, in the later you say, oh, that's too many genes assigned, or features assigned to the peaks. You can filter using like a distance, the shortest distance from a feature to the feature to the nearest start site distance, to filter later. As Jianhong pointed out, you can always look at the help menu. And you always are welcome to post a question in the support site, and we'll try our best to respond as quickly as we can.
About the track viewer, does it require to read the data into memory before plotted? Yes, but indeed, you can just read in subset of the data. You can use, there are multiple functions, import the data by the, you can define the plot region. For example, you define the plot region by a G-range and then import data. There are multiple functions in the track viewer to import data. Just import a subset of data. It will take only tiny memory to plot. Similar, like, is it able to query files on disk with annotation similar? Yes, anything can convert to G-range is okay for a track viewer to plot the lollipop plot. Yes, this is another thing I did not show here. You can plot the track viewer with multiple markers, for example, rectangle or arrow or labels. This is, you can assign it by some script. This can be used for automatically generate a batch of figures. For example, if you want to add arrow in the highest peak, it can be automatically generated for you. Can ChIP-seq annual package be used for DNA methylation data and annotation? I think the answer should be yes.
Yeah, I agree. Yeah, as long as you have the peak file, I think the ChIP-seq annual can handle, right?
So ChIP-seq annual can be used to annotate ATAC-seq data, ChIP-seq data, ChIP-seq data. So any type of data that has genomic ranges, any type of data that can be converted to genomic ranges. Basically, as long as you have chromosome location, chromosome name, and start and end, or even just have start and or peak site, and you'd be able to use that ChIP-seq annual for annotation.
For histone markers, ChIP-seq, what control do you prefer to subtract your actual signal for H3 or monk IP or input? It's fun to have one control for all the histone markers, or for each histone, a separate control is recommended. As I understand, if you have a monk IP, it will be much better. Otherwise, I think you prefer to use the input because it will sometimes people are using IgG or other things. The thing is that IgG is, there are several reasons for them to not use IgG. Is that because you have the IgG usually is prepared by different species as you prepare the antibody for histone markers. and it will pull down less genomic regions compared to your specific antibody. So it will not generate a good background for you. It will generate high duplicate rates and with low complexity. So it is not good. But the input will provide you more genomic coverage, and this is how say a lot of people are recommended use input or some monk IP, right? They band for different positions or different how say they just functions different from the antibody you prepared, and for is that we should have one control for all the histone markers, or for each histone separate controls is recommended. Yeah, this is indeed people are always suggest you to use multiple controls for each histone markers, but if you want to compare, I'll say when you do experiments, you'd better to prepare the controls for all your experimental. But if you want to compare different markers, compare the markers in different status, there is some debate about whether or not should we involve input as a control, because input control, some people think it will introduce some bias by the experimental. So how to say, depend on the algorithm you are using, you can select use input or not. For example, if you are using diff band, the author suggests you to use the input.
There is a question about duplicates.
Okay, wouldn't removing duplicate rates decrease the actual signal? I think for this question, this is for the pre-processing step. I think you should remove the duplicate rates. Otherwise, the signal can be best. So if you don't remove the duplicate rates, though the signal can be very high, but it's just best. So yeah, not sure whether that answered the question.
About the remove duplicates, there are two things. How say? Some software they will remove when they call peaks. They will do remove duplicates first. For example, Max 2. The first step is the remove duplicates, and then they will shift the rates in positive strain and negative strain, and then increase the signal background ratio, and then call peaks compared to the background. But some software, they did not have this step, so you want to remove it by yourself. And some software, they are using a more intelligent way to trace the duplicate rates. Sometimes they think this duplicate rate is not only come from PCR duplicates, but also maybe shows the true chip signals. So they have some model to feed the duplicates, whether or not should we remove it. So definitely when you call peaks, you'd better read the documentation of the peak callers and then decide whether or not should you remove duplicates. As I know for Max 2, I think you just mark the duplicates without remove it. It is good enough for you call peaks.
Yes, like Jianhong said, actually Max has a way to estimate whether the percentage or the number of duplicate makes sense. So that's they estimate what the duplication and percentage. So internally, they actually have this being automatically taken care of. And there's another question about track view support highlight in specific genome region. Have you answered that yet? I think yes, right? I remember.
Yeah, I answered that. We have a multiple
okay.
tools to add line in special positions, you can assign it by some script. So it is designed for high throughput script to generate the tracks.
All right, we are pretty much at the end of the session, and perhaps there is one more question. So how does ChIP-seq anode deal with this ambiguity?
Show this, you have really multiple parameters to control the annotation output. Here is a parameter called output. We can assign the nearest location, shortest distance, overlapping, or nearest bidirectional promoters. The result I shown here is what will output for the annotations. For example, there are two genes. If your site gap is 5K, right, gene C or gene B are all in the 5K. So if you set as the nearest bidirectional promoters, it will only output gene A and gene C for you. If you set overlapping, definitely it will output gene A, B, C. Did I answer your question?
So it looks like pretty much it gives you lots of flexibility, and depends on your biological question, you should select the parameters.
Yes.
All right. Well, this brings us to the conclusion of this session. Thank you all for great presentations, great questions.
Before we end, can I just say that for the histone import versus H3 versus those questions, I want to make sure that we don't get overstated. And so there is actually a paper they are compared this. So you might want to look. I just saw a paper. So the frontier in genetics, computational genomics, they compare different input, how they affect results. So you might want to, the person who asks this question might want to look into that instead of taking our answer, just because I think different questions might have, experimental condition might have different requirement. Just want to say that.
Like if you can share that paper in the chat.
Okay.
Yes.
Okay, sounds good.
All right. Thanks, everyone. That concludes the session.